Hey everybody, it's Dr. O. First of all, I hope everyone is safe and healthy. Obviously, these are not ideal times and an ideal situation that we're in right now, but what we're going to do is we're going to switch the class over to being fully online. It's not ideal. I don't like it very much, but we're going to do the best that we can with it. So at this point, it has been quite a while since we've actually met, and probably what we need most right now, since we're just finishing up the unit on the nervous system, we need a little bit of a review on the nervous system itself. So that's what the purpose of this video is here. So what we're looking at here is three different neurons. So we can go ahead and label these neurons here. So if we start with this one, we see just the little end of the axon, and we can see the axon terminal here. So we'll label this neuron number 1. And then we can see this neuron right here. We can actually see the entirety of this neuron, including its cell body, which is where its nucleus and all of its organelles and most of its cytoplasm are going to be located. Obviously, I hope you guys understand that neurons have dendrites that are located on the cell body. The only reason you're not seeing them drawn here is because, I've said this a million times to you, I am no good at drawing. I have no artistic ability, so do not take this drawing literally. This neuron should have dendrites. I just can't draw them. So we have what we'll call neuron number 2 here. And then all the way down here in the bottom right corner, we are seeing a third neuron, of which we can only see its cell body and presumably its dendrites. So we will call that neuron number 3, if I can finish off this circle here. There we go. Okay, so first thing, a little bit of terminology, right? So for any particular neuron, we'll stick with neuron number 2 here because it's the one that we can see everything of. We've already covered how neurons have a number of different structural features. So we've covered the cell body, which is where the nucleus, cytoplasm, and most of the organelles are. Dendrites, which obviously you can't see here. So we can go ahead and start labeling some of these things. So this would be the cell body, right? And then you have the axon, which is this very long process right here. And then we covered how not every axon, but most axons are going to be covered with a layer of insulation, which is called the myelin sheath. And we've covered the whole purpose of myelin is to speed up the conduction of action potentials. We covered how action potentials are conducted in unmyelinated versus myelinated neurons, and why you would expect it to be much, much, much faster with a myelinated axon. If you don't remember that, you might want to go back to our discussion on that. And then you have the axon terminal, which is where neurotransmitter is going to be released. Very important. And then one very important part of the axon, we've kept track of it because this is the place where if an action potential is going to happen, this is where it's going to occur. It's this region right here, which we are going to call the axon hillock. A couple of other names for it are the trigger zone, and then you will sometimes also see it called the axon initial segment. Okay, so at that point, this pretty much covers all the most important structural features of the neuron itself. So, little other bit of terminology here. So, what a neuron does is it synapses on either another neuron or some other effector cell, like a skeletal, smooth, or cardiac muscle cell, or some glandular cell. So when we talk about a neuron and its relationship to other cells that it is either communicating with or receiving communication from, we use a little bit of terminology to make sure that we understand which cell is which. So what we're going to do here is we are going to focus on neuron number 1 and neuron number 2. So neuron number 1 is synapsing on neuron number 2. So for this relationship, we would say that Neuron number 1 is the presynaptic cell, and neuron number 2 would here be postsynaptic. The idea being that this very narrow space that exists between the two cells, between the presynaptic and the postsynaptic cells, that is called the synapse. So the presynaptic cell exists before the synapse. That covers all the communication that happens before we get to the synapse. And then the postsynaptic cell is at the other end of the synapse. So it is receiving the communication. And another little something that you will remember is that when an action potential traveling down the length of an axon, we'll say down the axon of neuron number 1 here, when an action potential reaches the end of the axon terminal, we cannot propagate the electrical signal any further. So that's why we have to switch over to a chemical signal. So at this point, you may want to go back and review what triggers the release of neurotransmitter from the presynaptic cell. So you'll want to make sure that you remember the voltage-gated calcium channels and their role in releasing these neurotransmitter molecules. So these neurotransmitters, of which there are several different types, acetylcholine, norepinephrine, and dopamine are going to be the ones that we focus on throughout the semester. There are lots of others. Those are going to be our focus. So neurotransmitters are just signaling molecules. They're signaling molecules in the same way that hormones are signaling molecules. And what do you remember about what signaling molecules do? How do signaling molecules exert their functions? Well, they bind to receptors, right? So the receptors for these neurotransmitters, which are now in the synaptic cleft, these receptors exist on the cell body or dendrites of the postsynaptic cell. In this case, we're talking about neuron number 2. So these neurotransmitter molecules, after they have been released by the presynaptic cell, are going to bind to these neurotransmitter receptors. And neurotransmitter receptors are always going to do one thing. Neurotransmitter receptors are always going to open ion channels. So regardless of what neurotransmitter you've got, what neurotransmitter receptor that neurotransmitter is binding to, the end result is going to be an ion channel on the cell membrane of the postsynaptic cell is going to open. And we've talked about this before. What is it that happens to a cell in which you are moving ions in and out in either direction? You are going to get a change in the membrane potential. Remember that? So we spent a lot of time talking about how do you predict what kind of change in the membrane potential you're going to get based on which neurotransmitter you've got. And what we said is that it's impossible to know unless you know what kind of ion channel you're opening. Is it a sodium channel you're opening? Is it a potassium channel you're opening? Or is it something else? You've got to know what kind of ion channel is being opened if you're going to be able to predict whether you are going to change the membrane potential in one way or another. So we'll review that again here in just a minute. Okay, so at this point, these neurotransmitters are going to bind to their neurotransmitter receptors. We open ion channels. That is going to produce a very specific change in the membrane potential locally right at the point of entry for those ions, and we call that a graded potential. Graded potential. So a graded potential is a method of communicating over a distance, but here's the deal. A graded potential will get weaker and weaker and weaker the further those ions spread out and have to travel. So for that reason, it is not going to be ideal for us communicating over very long distances. That's what the action potential is for. So this graded potential, which comes in the form of either positive charges or negative charges, which have either entered or exited the cell at this point, these are going to start spreading out from their point of entry into the cell, from those ion channels. They're going to start spreading out throughout the rest of the cell body, and they are going to start migrating down here to the axon hillock. So all the graded potential exists to do is to eventually give us the action potential. Now, not every graded potential is going to give you an action potential. There are specific rules and regulations that we have to follow in determining whether we get an action potential or whether we do not. And again, we will cover that here in just a second. So at this point, we've got the graded potential. It is traveling towards the axon hillock. Let's take a break for just a second and review membrane potential. So the membrane potential for any given cell refers to the difference in electrical charges across the cell membrane. Now, you guys will remember that the inside of the cell for most cells in the body is going to be much more negative than the outside, relatively speaking. So we've drilled this over and over and over again in class. You guys, I'm sure, remember what your resting membrane potential is. If we plot it on this graph, it's going to be about negative 70 millivolts. So that is going to be our RMP, our resting membrane potential. So when a cell, like a neuron, is at rest, it has a membrane potential of negative 70 millivolts, meaning the inside of the cell is more negative than the outside. That is when the cell is at rest, meaning it has not been stimulated with any type of graded potential yet. So graded potentials come in two different forms, and we mentioned this just a couple of seconds ago. When that neurotransmitter opens those ion channels, the inside of the cell, depending on what kind of ion we're letting in or out, the inside is either going to get more positive, so more positive, meaning we go from negative 70 to negative 60, negative 50, and on up towards 0 and into the positives. We call that depolarization. Move this over here so you can see it better. Depolarization. And the type of graded potential that gives you depolarization is called an epsp, or a excitatory postsynaptic potential. If the graded potential coming from this neurotransmitter causes the inside to become even more negative than it already was, and keeping in mind it was already pretty negative, we call this hyperpolarization. And the type of graded potential that gives you hyperpolarization is called an ipsp, or an inhibitory postsynaptic potential. So you can infer from the names we are giving these graded potentials, one is good, excitatory, it's what you want if you want an action potential. The other being inhibitory, it's probably not quite what you want if you want the graded potential. But let's explore why that is. And to do that, we need to go back to the requirements for what does it take to give you an action potential. Okay, so if we look back at this action potential graph, a cell at rest is going to be at negative 70. A cell at rest at negative 70 is not going to be firing any action potentials. If we want an action potential, we need to change the membrane potential, and we need to reach a very specific threshold. That threshold, if you will recall, is right here at negative 55. I'm going to draw a little dotted line to indicate where threshold is. So that's negative 55 millivolts. That is our threshold value that we have to hit if we want to get an action potential. Now, where we are measuring the membrane potential for this graph is right here at the axon hillock. You can get very, very, very drastic changes to the membrane potential in the cell body, but that does not really matter to us. What matters to us is that we have to hit threshold here at the axon hillock. Getting any sort of other changes elsewhere is not really going to do it for us. Our requirement for getting an action potential, that graded potential has to spread to the axon hillock, and we have to depolarize the membrane at the axon hillock to at least -55 or above. Anything less than that will not give us an action potential. So, for that reason, depolarization, an epsp, that is the type of graded potential that we desire if we want an action potential, because a epsp is going to depolarize us and bring the membrane potential closer to threshold. An ipsp, on the other hand, is going to take us further away from threshold, and it's going to make an action potential that much more difficult to get. And you saw my cat just walk by. She's wondering what I'm doing here. Sorry about that. So epsp is what we want if we want the action potential. ipsp is not what we want if we want the action potential. But ipsps are useful if you want to keep a neuron quiet and you want to keep a certain pathway off and inhibited. Okay, so at this point, we've got our graded potential spreading out towards the axon hillock. Since we want the action potential, let's go ahead and assume it is an epsp that we've worked with. Most epsps are going to be caused by neurotransmitters binding to receptors and opening sodium channels. Sodium flows from high to low concentration from outside the cell to inside the cell. Positive charges coming in, the inside becomes less and less negative, more and more positive. That gives us depolarization. So let's say that we get to threshold here. We've got lots of sodium ions that have made their way to the axon hillock here, and that gets us to threshold here. So the very first event, once our epsp arrives, the very first event that gets us to threshold, that is going to open voltage-gated sodium channels. So these voltage-gated sodium channels that exist here on the axon but not anywhere else, these channels are going to open, and we're going to get a massive, massive, massive influx of sodium from the extracellular fluid into the axoplasm. And we're going to go up about as high as +30 there. We don't go any higher because at that point, you will recall, we inactivate those sodium channels so that they don't let any further sodium in. At about the same time, we also open voltage-gated potassium channels. Potassium is having a concentration gradient that is flipped compared to sodium. So if we open potassium channels, potassium is going to move from the intracellular fluid to the extracellular fluid, so the inside would become less and less positive, more and more negative. So that starts the repolarization phase. No more sodium coming in, potassium leaving. So we're going to let potassium out into the extracellular fluid. You will recall also that instead of going right back to the resting membrane potential, we're actually going to dip a little bit below that. And we owe that to the fact that those potassium channels are a little bit slow to close. So eventually we do close those potassium channels, but at this point we need to get back to the resting membrane potential value in order for us to be all rearing and ready to go for the next action potential that comes along. So at that point, we need to wait for the sodium-potassium pump to come along, get all that extra potassium back into the intracellular fluid, get any sodium back into the extracellular fluid, and that will bring us back to the resting value. And at that point, we have completed one action potential, and we should at this point be ready to go for a second one. So one thing that I'm not going to indicate here that you'll probably want to review is absolute and relative refractory periods. Basically what deals with when is it possible to stimulate a second action potential if the first action potential is not quite done yet? So you'll remember that the absolute refractory period, if the first action potential is in that refractory period, you have to wait until that's done before you can even think about starting a second one. Relative refractory period, it is possible to get another action potential. It's just going to be more difficult because you are still in this part of the action potential right here. You're hyperpolarized. The amount of depolarization it would take to get you to threshold is going to be a little bit higher than what it would be if you wait until you're back at rest right there. Okay, so at this point we have hit threshold here, and we are going to start stimulating an action potential one right after another until we conduct that action potential all the way down the length of the axon. So I have a myelinated axon here, so that action potential is going to appear to jump from nodes of Ranvier one after another. Okay, so that is saltatory conduction. One action potential happens at one node. We wait for sodium to move over to the next node to open more sodium channels, and those just happen one right after another until we get the action potential carrying all the way down, and we are going to release more neurotransmitter here, just like we did up here. So if you recall, neuron number 1 was the presynaptic cell compared to neuron number 2, presynaptic, postsynaptic. Our terminology is now going to have to change a little bit. Neuron number 2 right here is now the presynaptic cell to neuron number 3, which is now the postsynaptic cell. In this picture that I have drawn here, we have two synapses. There's one synapse, presynaptic, postsynaptic. There's our second synapse, presynaptic, postsynaptic. So everything is going to work very similarly here. We have neurotransmitter receptors here. They may be different neurotransmitter receptors than what we saw up here, but we are going to have receptors waiting for this neurotransmitter to come in, and when that neurotransmitter binds, we will open ion channels and we will get either an epsp or an iPSP here. So the last thing to really cover here in this little review that we've got going, when you're classifying neurotransmitters, if a neurotransmitter gives you an epsp, we call it an excitatory neurotransmitter. And if the neurotransmitter gives you an iPSP, we would instead call it an inhibitory neurotransmitter. So I hope you found this little review that I put together helpful. These are all things that we covered in lecture, so it shouldn't really be a surprise to hear some of these things. I do recommend going back and looking through the old lecture slides, looking through your notes, looking through the book, but this should be just kind of a quick and easy review. If you remember all of this stuff on first glance, that's great, you're in great shape, but it never hurts to look through these things again, especially since we're coming off of a 2-week hiatus. All right, so that does it for this video. Since we're going to be online for the foreseeable future, I am going to try to post lots and lots and lots of videos like this, so I hope you found this helpful. So for the time being, I will sign off, and I will see you again next time. Bye-bye.